Hi, I'm Ria Thomas. I'm a postdoctoral research fellow at the Neuroregeneration Institute at McLean Hospital. Today, I'm going to give you a brief overview about a project from our lab that got published recently. Broadly, the project focuses on lysosomal dysfunction in Parkinson's disease. So, Parkinson's disease, or PD, is a multifactorial neurodegenerative disorder, and several cell biological pathways that are dysfunctional in PD have been identified before. So, one such pathway that has been shown to play a role in the etiology of genetic and sporadic forms of PD is lysosomal dysfunction. And this particular project focuses on the role of a particular lysosomal enzyme called glucocerebrosidase and tries to identify the mechanism that leads to the reduction of this particular enzyme activity in idiopathic form of PD. So, glucocerebrosidase, or GCase, is encoded by the GBA1 gene, and it is a lysosomal hydrolase enzyme that is required for the metabolism of specific sphingolipids. And mutations in the GBA gene comprises one of the largest genetic risk factors for PD, and people with PD with this mutation exhibit reduced lysosomal GCase activity and accumulation of specific sphingolipid substrates. Now, what was interesting is that studies from our lab, as well as others, have shown that it's not just genetic forms of PD with the mutation in GBA that exhibit reduced lysosomal GCase activity, but also idiopathic or sporadic cases of PD also exhibit reduction in this particular enzyme activity. So idiopathic or sporadic cases of PD are the cases with no known genetic basis for the disease and comprise of majority of the cases of the disease. Almost 90% of the cases are idiopathic cases. So this was interesting, and towards this, the main aim of this particular study was to look at the mechanisms behind reduced lysosomal GCase activity in idiopathic form of PD. So for this study, the cellular system that we employed were fibroblasts isolated from human subjects. And we had three groups of fibroblast cells. We had the healthy subjects, fibroblasts from healthy subjects, which served as controls. We also had fibroblasts from two groups of PD, one from genetic PD, which had mutation in the GBA gene, and another group of fibroblasts isolated from idiopathic PD patients. So over to the results. This is how the GBA pathway looks in a healthy subject control. So the newly synthesized GBA protein gets post-translationally modified in the ER and the Golgi, after which it is trafficked to the final destination, which is lysosome, which is where it helps in the metabolism of specific lipids. So the GBA that is post-translationally modified in the ER forms a complex with another protein called LIMP2. So LIMP2 is a specific trafficking protein, and the complex of GBA and LIMP2 together moves from the ER to Golgi and then finally to the lysosome, which is its final destination. So now looking at the results that we observed in our cells, first in the genetic PD fibroblast cells, we mainly observed two phenotypes. One was that compared to the healthy subject controls, we observed that these cells exhibited increased retention of the protein in the endoplasmic reticulum, and they also exhibited reduced lysosomal GCase activity. So what is happening in these cells is that due to the presence of the mutation, the folding of the protein is being affected, thereby leading to increased retention of the protein in the ER and thereby reduced lysosomal GCase activity. And this is already known before, and this has been shown by other published studies in other cellular systems. So it was interesting that we were able to replicate the same things in our fibroblast cells as well. Now, the real interesting results actually came when we looked at the idiopathic PD fibroblast cells. What we saw in the idiopathic PD cells was that they also exhibit reduced lysosomal GCase activity and increased retention of the GBA protein in the ER, similar to the genetic form of PD. And in addition to this, these cells also exhibited a significant reduction in the level of the trafficking protein LIMP2. So for a quick summary of our study, of our key finding in this study, what we observe is that both the genetic and the idiopathic PD group of cells exhibit reduced lysosomal GCase activity, but it's the mechanism that lead to this deficit that seem to be different between the two groups of cells. In the genetic PD group of cells, it's the presence of the mutation that lead to increased retention of the protein in the ER and thereby reduced enzyme activity in the lysosome. And in the idiopathic PD case, our hypothesis is that it's the reduction in the LIMP2 level that's affecting the trafficking of the protein from the ER to the lysosome, thereby leading to reduced lysosomal GCase activity. So more broadly, such alterations in lysosomal enzyme activity and subsequent lysosomal dysfunction can lead to deficits in cellular degradation pathways, and such changes in more vulnerable cell types, such as the dopaminergic neurons, in the case of Parkinson's disease, could be detrimental and could eventually lead to their neurodegeneration. In terms of the significance of the study, I would like to point out two things. One, we have utilized fibroblasts in the study. So fibroblasts are more readily accessible cell types that are peripheral to the brain tissue. And it was interesting that we were able, fibroblasts mirror the same phenotype, at least in terms of GCase activity, as that observed in the brain tissues of idiopathic PD cells. And so fibroblasts could serve as good experimental platform for testing therapeutic paradigms aimed at manipulation of GCase levels. And the second thing is that in our study, we see that both the genetic and idiopathic PD, even though through different mechanisms, converge onto the same pathway, in this case, lysosomal GCase activity changes. And several therapeutic modalities aimed at improving lysosomal function, either by improving lysosomal enzyme activity or by reducing substrate accumulation, are already under development and could serve as interesting therapeutic targets, not just for genetic PD, but also for idiopathic Parkinson's disease. So that was for the summary of our study. Thank you very much for listening.